Hi, Dr. Wang here. So today we're going to do this assignment called PCR2. This used to be a exercise on paper when we're doing this in person. So in the virtual format, what I did is to provide you with the PDF file, and I think you can do highlight on PDF file. All right, so here, this is of course something that you can do with APE, but I think for the purpose of understanding the concept of restriction digestion and primer location, it's better to do this on paper because on this piece of paper, we have both the top strand and the bottom strand provided. So you get a more visual understanding of how these two techniques work. So we're going to do the first one. The first one is to find the restriction sites for the two enzymes. So here, restriction sites are palindrome sequence, which means you only need to read the top strand. You don't have to look at both top and bottom. You just scan the top strand looking for GAATTC. So I'm going to do a demo here because I can see GAATTC. So I'm going to highlight, and it's right here, GAATTC. Is right here. And then I'm going to use a red marker to mark the location of the cut. It cuts in between G and A, right? So I'm going to cut like right here on the top strand. Now I'm going to zoom in so that you can see this a little bit better. So GAATTC, the enzyme always cut identical location. So at the bottom strand, it's also GAATTC from 5 to 3, and it cuts just like the top strand between the G and the A. So it cuts right here. So the question asks you to provide the location for the location of the cut. So for the top strand, you are going to provide the location of this G right here. So how do you know the location of the G? All the sequences are numbered. So here, this is 50, right? So 50 base pair right here. And then here, 60 base pair. Here, 70, right? So you can see this larger mark. So this is 10 base at a time. So the second question will ask you to provide the location of the bottom strand. So then you're going to provide the location for this guy that's right here. All right, so this is how to do equal R1. And then the same thing for HPA1, you're first going to scan the top strand looking for GTTAAC. So just here, remember that here, the cut is right here. So where does it cut on the bottom strand? Remember, it always cuts between GTT AAC, between the middle T and the middle A. That's where it cuts on the bottom strand. All right, so this is how to solve the restriction enzyme related question. So now let's figure out how to find the primers. So here, let me clear this a little bit so we have a fresh start. Okay, all right. So primer is a little difficult to find because for primer, you will have to scan both the top strand and the bottom strand. So I can actually tell you that you only need to scan this one and this one. So the three primers are either located over here, right? This is a continuous DNA molecule, but the primers are not in the middle. They're either at the beginning or at the end. So this way you don't have to scan the two in the middle. This is going to sort of make it a bit easier for you. So let's see, let's just scan one for an example, right? So we're looking for TTTC CGA, right? So I'm going to do that first. I'm going to scan the top strand first. If I don't find it on the top strand, if I scanned from here to here and from here to here, I just don't see it, that means it's not on the top strand. Then you need to scan the bottom strand. The way to scan bottom strand is actually to go in reverse because all the primer sequences are provided as 5 to 3. So when you scan, you have to scan from 5 to 3, right? So that means if you're reading the top strand, you're going to scan them this way. If you're reading the bottom strand, you're going to read from 5 to 3. Right, so let's scan the first one. So TTTCGA, and I can see that actually it's right here, right? So CGA, and it ends with TACTT. TACTT is right here. Okay, so this is where the primer sequence is. So as we can see that this primer is going this way. So is it the forward primer or is it the reverse primer, right? One of the questions will be asking you this information. So if the primer is going this way, what about if you find it on the bottom strand there? So for example, if we discovered a primer sequence that's going this way, then is it forward or reverse? And finally, for a PCR to work, you have to find a pair of primer that's going opposite towards each other. One forward, one reverse, going towards each other.